Okay. So, very good evening to all of you. So, I'm going to start today with a topic called biotechnology and its application. Biotechnology and its application. And we are to continue from humulin. I think last day we have completed a little bit of humulin. Have we? Okay. So, we are going to start from gene therapy. Fine. Basic idea about humulin again, because it was the last topic on the last very day. So we will start with humulin first, and then we are going to complete gene therapy. And I think we are going to complete the topic today itself. We are going to complete the chapter. Okay? So let us start with the topic called biotechnology and its application. Okay. Now, in this topic called humulin, we have talked about in humulin. What is humulin? Humulin is human insulin. Humulin is human insulin. Now, where from insulin is being secreted? It is secreted from the beta cells of islets of Langerhans. What are islets? Islets are islands. Who is Langerhans? Is the scientist. The name of the scientist. Islets of Langerhans. Fine. Now, this insulin is administered to the people who are suffering from diabetes mellitus. That means blood sugar. And it happens why this blood sugar increases? Due to the inactivity of insulin or the less secretion of insulin. So the beta cells of islets of Langerhans, if in any ways secretes less of insulin, the function of insulin is to make glycogen from glucose. That means what it does is it converts glucose into glycogen. But what you have to know is inside the bloodstream, if you have got huge amount of glucose inside the bloodstream, if you have got huge amount of glucose, this is glucose. They are certain, and if this glucose needs to get inside the cells of your body in order to get what? The tissues, the cells, they need what? They need glucose, and they need glucose to get energy, and they are going to perform respiration, aerobic respiration, and they will break down this glucose into carbon dioxide, 6 molecules of water plus 686 kilocalorie of energy, which is equal to, which corresponds to 38 ATP. So the cell in order to get this particular glucose, energy, they need to break down the glucose. But how come these particular cells will be getting glucose from the bloodstream? There is a particular, if you remember in the biological topic, there are some receptors called GLUT4. GLUT4 is a receptor which can help the entry of the glucose inside the cell. But for that entry, the presence of insulin is going to open up the GLUT4. It's going to help in the opening of the GLUT4 so that this particular glucose can get inside the cell. So glucose can only get inside cell if there is a presence of insulin, because insulin is going to activate GLUT4 receptors so that the glucose can enter inside the cells. And if the glucose enters inside the cells, ultimately what is going to happen? Your bloodstream is going to have less of glucose. Got the point? So that is our purpose. But if there is problem in the formation of insulin, if there is less of insulin, then the glucose uptake will be less. The glucose amount is going to increase. If the glucose amount increases inside the bloodstream, we call the condition as diabetes mellitus. Fine? Now, so we need insulin, and this insulin has to be injected inside this particular patient suffering from diabetes insipidus. So before biotechnology came into place, we used to depend upon slaughtered cattle and pig. So slaughtered cattle and pig के ऊपर depend करना होता था for insulin. And this insulin which we got from cattle and pig, definitely we are going to get the effects of the insulin, But along with that, we are going to have unwanted LRTs. When biology came into play, but before biology, let me tell you, the name of the scientist named Banting and Best. Banting and Best. They got Nobel Prize. They got Nobel Prize because they got a solution. Of what? They got Nobel Prize. They got a solution. And they got this particular insulin. They derived this insulin from a dog. And they got Nobel Prize for that. So insulin was discovered by Banting and Best. Banting and Best. But before biology came into play, we depended on them. Cattle and pig, slaughtered cattle and pig. But when we got the idea about plasmid, we got the idea about plasmid, we got to plasmid of what? Which organism? E. coli. So we have cut the particular E. coli by the help of what? By the help of restriction endonuclease enzyme. Say for example, what is the name? One of the example of REN is ECOR1. And we have talked about different types of restriction endonuclease enzyme. And just help me, Pratyasha, some of the examples of clavon, PBU1, PBU2, then HIN3, then name some damage1, SAL1, good. PBU2, we have talked about that, SAL1, PST1, ECOR1, clavon. Okay, so these are the different types of restriction endonuclease enzymes. So by the help of this enzyme, we can always cut the DNA in their specific site. And what is that site called? Palindromic site. What is that site called? Palindromic site. Any issues with this? Come on. Great. Now, the moment we have cut it down, cut this particular portion, we need to add the gene of interest. What is the gene of interest? What is the gene of interest? What is insulin? It's a protein. It's a polypeptide chain. It's a polypeptide chain of amino acid. It's a polypeptide chain of amino acid. But how many polypeptide chains are there? Chain A is there, chain B is there, and chain C is there. So this is chain B and chain C is there. And chain C is there. This is C. Okay? So chain A is having 21 amino acids. Chain B is having 30 amino acids. 30 amino acids. So it's a polypeptide chain of amino acid. C is the linker between A and B. So they are being produced normally by what? Polypeptide chain. That means the chain of amino acids are always produced from the DNA. So how on earth we are going to understand which portion of the DNA makes insulin? Come on, we are already blessed with HGP, Human Genome Project. Yes, we know exactly which part of the DNA can give rise to what? Can give rise to the insulin forming DNA. So we are going to cut those sections of the DNA which can make insulin, and this particular rDNA, now we are going to put it back inside the E. coli bacteria. Fine? And then what we can do, can we take the whole of the DNA which is going to make A, C and B together? No. A, B and C, these polypeptide chain together makes it an inactive one called proinsulin. If you just get back to the basic cellular structure, remember this is central dogma. This is central dogma, and you know what? DNA makes its copy called by DNA replication. Then what is being formed? mRNA by the process of transcription. This is very unique to you. You know it already. We are in the verge of finishing up all this concept. Fine, it's already over. mRNA to protein is by the process of translation. This we. Come on, who will tell me who discovered this? Central dogma was given by Francis Crick. Very good, Francis Crick. Now, this is what? This protein is produced where inside? Ribosome, the larger subunit and smaller subunit, and this is the particular mRNA. 5' to 3'. Fine, this is very good. This is P site and this is A site. This P site and A site. Think about it. Okay. Technical problem. Yes. P site and A site. Okay, let me tell you. Now, P site and A site. So there are two sites in ribosome. There are two sites in ribosome. There are two sites in ribosome. And remember, two sites. What is the meaning of the term P site? Peptidyl site. And what is A site called? Amino acid site. So ultimately they are going to help in what? tRNA is going to come, transfer RNA is going to come and start forming what? Start forming. It's going to have what? It's having four sites. tRNA is having four sites. They are also called as adapter molecule. They are also called as adapter molecule. In adapter molecule, they have got four sites. At the 5' end, they have got, at the 3' end, they have got amino acid binding site. Then they have got anticodon site just opposite to it. Then at the right side they have got DHU loop. And the left side they have got T-IC loop. Fine. Amino acid binding site and then ribosomal binding site, if you remember it. And they are going to come up with, in the P side they are going to come up with one amino acid. They are going to come up with one amino acid. They are going to come up with one amino acid. And ultimately is going to make what? If this is the ribosome, this is the ribosome having P site and A site. P site and A site. This is P site and A site. And remember, these are codons, and the tRNA which is going to come over here is going to have this anticodon site. Fine. If this is AUG, they're going to have what? UAC, and it's going to contain what? Methionine. And if this is CGU, the tRNA should be containing what in the anticodon site? GCA. Say it's having valine at the anticodon site, at the amino acid binding site, and translocation is going to occur. So it's going to make what? A chain of amino acid. It's going to make a chain of amino acid, and this chain of amino acid which is produced inside inside ribosomes is ultimately being formed by the particular DNA. Now this particular amino acid produced by the ribosome, do you think it is going to straight away come out of the cell, or this particular protein now needs to get inside some other cell organelle which is going to help in the endocytosis? Think about the cell chapter. Do you remember about endoplasmic reticulum? Do you remember about the three types of the structures called tubules, cisternae and vesicles? Do you remember about the functions of Golgi body which helps in modification, packaging, glycosylation? Remember that? That means consider this to be the proinsulin. Consider this to be a proinsulin with A polypeptide chain. In between what is there? C. And again, B. What is this? B. This is also polypeptide chain. Clear? Now this is the proinsulin. This is the proinsulin. It reaches where? In the endoplasmic reticulum first. Endoplasmic reticulum is going to check it out whether it is having formed okay or not, and it's going to pack it, send it to the Golgi body. Golgi body is going to go for what? It's going to modify it. What is the modification? It's going to keep on the A and throw away, cut off the C and make what? B. A and B together, they make a disulfide bond and make what? Make insulin. Make what? Insulin. This packaging is done by the Golgi body. A is having 21 amino acid, B is having 31 amino acid. So C is being removed. This is a part of the Golgi body which is removed. The part of the Golgi body which is removed by the part of the proinsulin is removed by Golgi body. This is one type of modification. If this happens, if this insulin is an active insulin, we can always, by the help of biotechnology, we can always take this particular A gene which makes this polypeptide chain, and we can introduce the A in the plasmid, the A polypeptide forming DNA, and we can insert it inside E. coli bacteria. This is A peptide forming DNA, polypeptide forming DNA, forming DNA. Is this clear? And we have put it inside the E. coli bacteria and made it what? Our DNA put it back inside the bacteria, E. coli, and allowed it to grow by giving it nutritive medium, and then we kept it inside bioreactors. Inside? Bioreactors. And found what kind of chain? Only A polypeptide chain. So what we have got? We have got billion copies of only A polypeptide chain by the help of E. coli. So what we have done over here? We have done genetic engineering, and we have found only A polypeptide chain. Do you think only A polypeptide chain is going to give us insulin, or we have to also do the same with B polypeptide chain? So we have to do separately. We have to make this B polypeptide chain, put it inside another plasmid, we have taken the gene which can only form B polypeptide chain, and we can introduce this RNA inside the bacteria, E. coli bacteria, and we know the process how we are going to do it. Fine, so we are going to get billion copies of only B polypeptide chain, and then what? In vitro, what we are going to make is we are going to make a disulfide bond between A and B and make the human insulin, which is called as humulin. This humulin was made by the American company for the first time. The name of the company is Eli Lilly. It's an American company in the year 1983. 1983. So American company, Eli Lilly. Mind you, it's very, very important for board exam and also very important for your NEET examination. Is this clear? So lastly, we have talked about this. Student, what is the addition? The addition is all the cellular aspects. So we have talked about that. Clear? Now, the most important is the next one called gene therapy. Gene therapy. And what is gene therapy? Mind you one thing. What is gene therapy? So this is a new term. It's a new term called gene therapy. Who is going to tell me the examples of autosomal recessive disease? Tell me some of the examples of autosomal recessive disease. Traits. Autosomal recessive traits. Come on.
Sickle cell.
Very good. Sickle cell anemia. Hemophilia. Do you think hemophilia is? It's a sex-linked disease. Phenylketonuria. Very good. Then you're missing one. Most important. Thalassemia. Albinism also comes under this. These are examples of autosomal recessive. Just add one other one called ADA deficiency. ADA deficiency comes under autosomal problem. It's an autosomal recessive trait. Today we are going to talk about the last one called ADA deficiency. So gene therapy, in this topic gene therapy, we are going to talk about a problem called SCID. We'll talk about a disease called SCID. What is it? What's the full form of SCID? SCID means severe combined immunodeficiency. Severe combined immunodeficiency disease. Severe combined immunodeficiency disease. Severe combined immunodeficiency disease. Fine. Now tell me, what is deficient? Immune system is deficient. That means the person suffering from severe combined immunodeficiency is definitely going to become vulnerable or susceptible towards different types of disease. Correct. So this person is going to become susceptible towards different types of disease. So SCID is close to AIDS. Fine. In AIDS also what happens? Acquired immunodeficiency syndrome. What is deficient? Immune system. Who makes? Tell me the cells which makes the immune system of your body. B lymphocytes and T lymphocytes. B lymphocytes and B lymphocytes and T lymphocytes. Alito, tell me. B lymphocytes and T lymphocytes. They help in what? Humoral immunity or cell-mediated immunity? B lymphocytes humoral and T lymphocytes cell-mediated. Now these two together they make what? Together they make the immune system. So for India they are the soldiers. For India, very good thing. If you have got good amount of soldiers, We can also talk about this, that the total GDP of Pakistan, that they spend more of this money in what? In defense system. So that is good in one way, but bad in other ways. Fine, they're not going to take care of the education, so they don't have. So now think about it. If you have good number of what? Lymphocyte cells, it's good for your body. It protects your body. But always remember, too much of everything is sometimes bad. Too much of something is bad, because there is a feedback mechanism. We're going to talk about that today itself. Is this clear? Okay. Okay, mosquito is running the show. What is going on? This one. Okay, tell me, it comes under which order? It's having two pairs of wings. Diptera. So, ये याद कराने के लिए ये आया था. Okay, so now, clear? B lymphocytes and T lymphocytes. B lymphocytes and T lymphocytes. They are the main. They increase the immunity of the body. Now let us talk about this. See, tell me what is this? Tell me what is this? This is base. This is pentose sugar. This is pentose sugar. Carbon 1, carbon 2, carbon 3, carbon 4, and this is CH2OH. This is CH2OH. What is this? This is ribose sugar. And this is narinous base. This is NB, narinous base. These two together makes what? These two together makes nucleoside or nucleotide? Good. This two makes nucleoside. So narinous base plus sugar makes nucleoside. What you can add and make it tight? You can add phosphate at the fifth carbon of the pentose sugar to make it nucleotide. Nucleotide. Any issues with this? Great. Now, the moment you add phosphate, now it becomes tied. Now it becomes nucleotide. Pentose sugar is having OH over here and OH over here. So this becomes what? Ribonucleotide. Because what kind of sugar is this now? Ribose sugar or deoxyribose?
Right.
What is the difference between, I think you know all this. What is the difference between, the major difference between deoxyribose and ribose sugar? Presence of oxygen in the second carbon. In DNA, in deoxyribose sugar, this second carbon is not having one oxygen. So this is ribonucleotide. This is ribonucleotide. Fine. Now, think about it. There is an enzyme called ribonucleotide reductase. Ribonucleotide reductase enzyme. So what is it going to do? This is going to change. This was what? OH. This is going to change. This ribonucleotide will be changed into what? This is base. This is narinous base. And say this is the pentose sugar. This is the pentose sugar. And second carbon. This is the third carbon. This is fourth carbon. This is the fifth carbon. CH2OH. And it's going to be the phosphate. Now, what happens? OH is present in the third carbon. And the moment reductase enzyme is being added, ribonucleotide reductase is going to change it into only hydrogen. So this becomes what? This becomes deoxynucleotide. This is going to become deoxynucleotide or ribonucleotide. Okay, fine? Now let us consider this narinous base to be. So narinous base can be of two different types. Purines or pyrimidines. Purines and pyrimidines. So purines are adenine, guanine. Purines are adenine and guanine. So think about it. If we consider this narinous base to be adenine, then think about it. This is what? Ribose sugar. This is ribose sugar. Sorry, deoxyribose sugar now. H. This is OH. And what is present over here? Adenine is present over here. In adenine, what is present? Do you remember the structure of adenine? Fine. It's having amino group over here. Fine. This was the structure. Imidazole group. This is the bond. Fine. This is amino group, and this is CH2OH. This is CH2OH, fifth carbon, and this is phosphate. This is what? This is what? Deoxy adenosine. Deoxy adenosine. If you remove this phosphate, this becomes what? Deoxy adenosine. Now, this amino group, the presence of this deoxy adenosine at huge amount, presence of this amino group, presence of this high amount of amino group inside the body is going to act as toxic substance. It's going to act as toxin. It's going to act as toxin. It's going to act as toxin. So there is a need to remove this amino group. What is the process of removing amino group? Deamination. And who is going to do this? If inside your body there is ADA, presence of ADA. ADA means adenosine deaminase enzyme. If you have this enzyme in your body, this enzyme removes this amino group and helps in deamination of this amino group. This is good for you, bad for you?
Not really.
Why not really? Because if it remains inside your body, then it is going to act as a toxin. So ADA, the presence of ADA gene, ADA enzyme, this is enzyme. This enzyme is there protein or non-protein? Protein. And this protein is produced from what? mRNA. mRNA is produced from what? DNA. Is this clear? So there must be a DNA, which means there must be a DNA, or there must be a gene which is going to make what? mRNA. And that mRNA should make a protein called adenosine deaminase. It's a proteinaceous in nature. Any issues with this? Come on. Now, what is going to happen? If there is a deficiency of this, the amount of deoxyadenosine is going to increase in your body or decrease in your body? Again, I repeat this question. If there has been a deficiency of ADA, this enzyme is not there. So it's not going to be able to remove amino group. In that case, your body is going to become toxic, number one. Clear? If it increases more, if there has been deficiency of ADA gene, the gene which can produce this enzyme, if there has been a deficiency of this ADA gene, then what is going to happen to the content of this? It's going to increase or decrease? Increase. If this increases, then by the feedback mechanism, it is going to stop the functioning of this enzyme also. It is going to stop the function of this enzyme also. Ball bujlam na. Pratyasha, tell me, what I have talked about right now? ADA gene, less of ADA gene, less of ADA, less of ADA, less of that enzyme, deamination won't take place. Deamination won't take place. Toxicity is going to occur inside your body, number one. Number two, if this increases, if the amount of this increases, it shows a negative feedback to this enzyme called ribonucleotide reductase. If this is less, do you think you are going to have good amount of deoxynucleotide? And if you have less amount of deoxynucleotide, think about your whole body runs on what? Gene, DNA. And what is the base of the DNA? Nitrogenous base. So one of them is purine. Got the idea what the deficiency of ADA can do. But in some patients, ADA gene deficiency is found. And when the ADA gene deficiency occurs, what happens? What happens? The X and T lymphocytes number gets it has got a strong effect on B lymphocytes and T lymphocytes. So ADA gene basically helps this B lymphocytes produced from the bone marrow, T lymphocytes produced from thymus gland, you know that. So the presence of this ADA gene, it can make these two effective. If there is a less of ADA gene, then definitely the enzyme is going to be less, and what is going to happen to these lymphocyte cells? They are going to perform less, and if the soldiers performs less, your body is going to become vulnerable towards the weak. Very good. Vulnerable towards attack by different types of pathogen. Different types of pathogen. Is this clear? So, clear? Very good. So, what is that particular problem if ADA gene is less in the body? The problem that just now we have talked about is about scale. So, how to solve this problem? So, this gene is sex-linked or autosomal one? Autosomal one. So, we have got two steps, two ways we can solve this problem. First, take ADA gene in a micro injection. Put it inside this particular baby who is suffering from this. Baby or adult? Okay. Baby. Inject it inside the baby. Inject this inside the baby. Come on. You have taken this ADA enzyme, ADA gene, and you have introduced ADA gene. Huh? Okay. And you have injected this inside. This is the ADA gene, and you have injected inside the baby. This concept is called enzyme replacement theory. Enzyme replacement concept. Enzyme replacement therapy. Enzyme replacement therapy. Do you think this is going to work?
So, inject enzyme now.
I'm injecting gene, and that is going to make the enzyme. Think about the size of the injection. You are introducing inside the particular body, and do you think this is going to work? Shesta, tell me. A wild guess. Gene introduced করে দিলি body মধ্যে baby ভাবলো baby body ভাবলো this is antigen and you start killing them because it is not going to last for long.
Sir, gene টা তো
जीन অন্য जीन के केटे ढुके जाবে. This gene is going to cut another segment of the DNA जहाँ पे उसका deficiency है वहाँ पे वो ढूंढेगा क्योंकि वो biology जानता है, वो genetic material जानता है, it knows what is human genome project, knows about কেন হবে না?
কিন্তু ওর বডি তো প্রোটেকশন নেই তো যে ওকে করতে পারবে
কি করতে পারবে? You are just placing ADA gene inside the body and you want it's going to last long, it's going to get degraded. নষ্ট হয়ে যাবে.
বারবার দিলেই হয়ে যাবে.
So বারবার দিতে হবে. Great. বারবার দিতে হবে. So it's a temporary solution. It's a temporary solution. बार-बार दिते होबे. Rightly said. So आपको बार-बार देना पड़ेगा ये injection. You need to introduce this particular enzyme on a regular basis. So enzyme replacement theory is therapy is not going to be the.
Sir, একটা ইনজেকশনের কত দাম হতে পারে?
I don't have any idea, but it's going to be costly. Clear? So enzyme replacement therapy fails. It fails. It's not going to be a permanent solution. So what is the other term? Gene therapy is the solution. But do you think gene therapy is also going to be a solution? Let us find it out whether this becomes a solution or not.
Injection body প্রত্যেকটা সেলের মধ্যে ইন্ডিভিজুয়ালি
Good. Very good. Very good. Very good. Very good.
সেটা তো পারবে
না হবে না. So you're going to just inject in the body.
আচ্ছা যদি তারা নাই পারে তাহলে ওটা মানে সেল মেমব্রেন পাস করে রাইবোসোম খোঁজার পর নিজে রেপ্লিকেট কিছু করে নেবে
খুব করবে but ineffective. অত বেশি ভালো করবে না. Rightly said. Good thought. Clear?
I will try.
Good. Now, all of you. Done? Sure. What is gene therapy then? So there is the concept of what? Now comes the gene therapy. For this, we are going to use retrovirus. What is retrovirus? Retrovirus is what? RNA virus. So,
17 crore একটা ডোজের জন্য
লাইফ টাইম চলবে 17 করোড়
enzyme replacement therapy.
সিঙ্গেল ডোজ বানিয়ে কিনতে পারবে
Great. So you have to become billionaire. So what we have found out, for the injection to be given on the body, the enzyme replacement therapy, it costs 17 crore rupees.
ও
According to Google. Huh?
জল জেন্সমা বলে একটা আছে ওটা ওয়ান টাইম জিন থেরাপি ট্রিটমেন্ট তার আর লাগবে না ওটা 17
Biotechnology.
আচ্ছা আচ্ছা
17 crores.
বাপরে
Okay. That is
Now
gene
to
therapy.
70 crore.
Success rate-
Great.
success rate
Very good. So
হচ্ছে
Google
99%.
helps you like that. Okay. Retrovirus. So
So
virus having RNA.
Guys,
Retrovirus is virus having RNA as genetic material. Virus having RNA as genetic material. Now think about it. What RNA does is when this virus infects a normal cells. Say for example, this virus infects the lymphocyte cells. Infects the lymphocyte cells. What it is going to introduce inside the lymphocyte cells? Tell me. Deepti. Retrovirus. वो RNA lymphocyte में देगा या DNA देगा?
RNA देखो।
RNA देगा. Think about the retrovirus called AIDS. We have already covered AIDS. AIDS में it is going to introduce RNA. So if this is a cell, it is going to introduce the RNA, and this RNA by reverse transcription is going to make what? Double stranded DNA. It's going to make what? Double stranded DNA. So what is this called? Reverse transcription. Say for example, this is the particular DNA, this is the particular gene, and this portion of the DNA, this portion of the DNA, this portion of the DNA can give rise to ADA. This portion of this is the ADA gene. This is the ADA gene. So you have cut this portion of the ADA gene, introduce this inside the retrovirus. Clear? The moment you introduce this ADA gene inside retrovirus, retrovirus के body में cell के अंदर क्या रहेंगे अभी? ADA gene. So when it is going to infect lymphocyte cells, now why lymphocyte cells? Because from that person who is suffering from SCID, you have taken out from the bone marrow, you have taken out lymphocyte cells. You have taken out lymphocyte cells. बोलो इस lymphocyte cells के अंदर क्या ADA gene है? The person who is suffering from SCID, you are taking out the lymphocyte cells from the bone marrow. Now tell me, these are the cells of the bone marrow. अब ये cells के अंदर क्या ADA gene है? Is this present or it's not present? Not present. So it's not present. ADA gene is absent. ADA gene is absent. ADA gene is absent. So what you can do is you can take this ADA gene, cut this ADA gene from the normal healthy person, and that person is going to be close relative or distant. Why?
because যাতে অ্যান্টিজেন হিসেবে না কাজ করে
Very good.
ঠিক বল
Remember organ transplantation?
হ্যাঁ
Organ transplantation, there is a term called HLA.
HLA.
So you're not going to activate HLA. So you have to take that ADA gene from close relative of the person who is suffering from SCID.
স্যার
Is this clear? Huh? Mhm.
মানে একটু সাইড লাইক আমার একটা রিলেটিভের কিডনি পুরো ফেলিয়ার হয়ে গেছে দুটো কিডনি
Mhm.
তো ওনার ওয়াইফ আছে
হুম।
ওয়াইফ বলছে একটা কিডনি দেবে
No.
Is it possible?
Why not possible? Possible. Possible.
ম্যাচ
But
তো নাও করতে পারে
histoincompatibility, the histoincompatibility chances are going to be more or less.
Less.
Blood relation আছে?
No.
No. Clear? Even they are having the same blood group, but still there will be histoincompatibility. What is the meaning of histoincompatibility? Histo means what? Tissues. So there will be incompatibleness with the tissues. So cyclosporine A has to be administered, but in higher doses, because incompatibility will be more or less. But with the same relation, mother giving kidney to son,
লেস
less. Though the blood group can be different, but still some of the affinity, some of the sameness, some of the commonness will be there. Got the point? Now, so ADA gene from the close relative will be taken, introduced inside RDNA. RDNA नहीं जानता है उसके पास क्या है, हम जानते हैं उसके पास क्या है. We have done it. Retrovirus को लिया, उसके अंदर ADA gene को घुसेड़ दिया, in fact inserted it, and allowed the RNA virus to infect these cells having no ADA gene. So when a retrovirus is going to make this RNA, we are going to call this as cRNA. What is this cRNA called? cRNA. cDNA. cDNA. Why? Because it's going to make what? It's going to make RNA from. It's a retrovirus. So retrovirus is going to make DNA by reverse transcription. So that is called as cDNA. That is called as cDNA. And this cDNA contains the ADA gene, and that it is being injected where? We have allowed the retrovirus to infect on the lymphocyte cells which are not having adagene. So it's going to introduce what? The cDNA inside it. Now this lymphocyte cells will be allowed to grow in vitro. We're going to help them grow. Just like in tissue culture. We're going to help them to grow. And put this lymphocytes loaded with adagene back inside the bone marrow. Back inside the bone marrow. Got the point? So now the bone marrow, because they are stem cells. Stem cells. Bone marrow contains what? Stem cells. So you have done this. You have done this genetic engineering or gene therapy with the stem cells of the bone marrow cells and introduced your adagene by the help of retrovirus. Got the point? And then inside the body they are going to create what? As normal lymphocyte cells with what? ADA or no ADA? So ADA है तो ये अदाएं दिखाएंगे नहीं दिखाएंगे अदाएं? So they are going to show some अदाएं. Good. अदाएं और अदा आगे दिया. Now ये ना बोल. Adagene you have got it. Introduce this adagene inside retrovirus. Retrovirus is having RNA. RNA is going to make DNA. That DNA will be called as cDNA. So cDNA of adagene you have created. Then this retrovirus having cDNA, you have allowed the retrovirus to infect the normal cells of the person who is suffering from SCID. Normal cells means lymphocyte cells, which is not having adagene, adagene. Now, the moment retrovirus is going to infect, is going to insert the cDNA inside the lymphocyte cells. Then, these lymphocyte cells now, they are loaded with what? ADA. They are happy. And they are going to show ADA. And you are going to help them increase in number by giving them some liquid medium. The lymphocyte cells are going to grow, and then you have to take these lymphocyte cells having adagene back inside the bone marrow, because bone marrow contains stem cells. If I ask you these questions, try to answer me. What is the stem cell of RBC? Hemocytoblast. What is the stem cell of WBC? Myeloblast. What is the stem cells of platelets? Class 11. Megakaryocytes. Think about these are all stem cells. So stem cells are the original copy. तो original copy से क्या बनेगा बाबा? Copy बनेगा? आप photocopy बना रहे हो तो उसका क्या होगा? Replication होगा तो क्या बनाएगा? Adagene बनाएगा? Adagene is going to make what? ADA is an enzyme. It's going to make what? It's going to deaminase. It's going to help in deaminase of that adenosine, deoxyadenosine. तो बोलो अच्छा होगा आपके लिए खराब होगा? B lymphocytes are going to get loaded with what? They are going to start fighting back and it's going to protect your body from different pathogens. Any issues with this? Because the function of deoxyadenosine is what? If it forms in more content, it has got two effects. First, it's going to stop the activity of the lymphocyte cells. Second, it's going to stop the function of ribonucleotide reductase enzyme. Remember that? So this is how the gene therapy is being done. Ab bolo, there are two things. Our body, there are two types of cells. One is somatic cells, and the one is germ cells. Somatic cells, and the one is germ cells. Germ cells. Somatic cells means normal cells. Now, if you are going to do this in the somatic cells, you have to regularly do it. A couple having the first child, I think we have talked about this in reproductive health topic. Remember, we have talked about karyotyping. Karyotyping, by the help of karyotyping, gene mapping, this is called gene mapping. You can find out if the particular baby who is there inside the mother's uterus, the fetus, is having, whether the fetus is having any genetic problem or not in the form of Klinefelter syndrome or this and that. Remember that? We can always check it out today. And if you find that this particular baby is having ADA deficiency, if you find that this particular baby is having ADA deficiency, so बोलो किसको इंजेक्ट करेंगे? फादर को इंजेक्ट करेंगे? ये प्रोसेस किसके साथ होगा? फादर के साथ, बच्चे के साथ? Aditya is going to tell me. The baby is suffering from ADA deficiency. So किसको इंजेक्ट किया जाएगा? मतलब ADA process, the gene therapy will be done with father or the baby? Baby. No. Bhavar karon ache. Baby. I will tell you. Clear? Because these are somatic cells. But baby में ये क्या sure shot हो जाएगा? Inside baby, do you think it's going to be sure shot? ये baby अभी बड़ा हो गया, बाप बन गया.
बाप को भी देना पड़ेगा.
फिर से बेबी को देना पड़ेगा.
नहीं, बाप को देना पड़ेगा.
अब बोलो, ये बेबी अभी बाप बन गया. तो जब बाप बन गया तो उसका germ cells है नहीं है? तो germ cells में if we introduce the same thing, ADA gene therapy, if we perform in the germ cells, it is going to be a solution. Next, बच्चे में ये नहीं आएगा. बोलो समझ में आया कि नहीं आया?
মানে ফার্স্টে যার হইছে ডেফিশিয়েন্সি আডা তাকে দিলাম, দেন ও যখন ফাদার হয়ে গেল তখন ও জার্ম সেলসে দিলাম.
হ্যাঁ, কারণ ওর থাকবেই.
হ্যাঁ.
And this is autosomal recessive, mind you that. Autosomal recessive means somatic cells are not going to have it. Fine? So think about it. You are going to get the best results if you perform this in the germ cells. No, if you perform that with the germ cells, high ratio नहीं करना पड़ेगा. But mind you, you have to perform all this in which kind of body? Embryonic cells. You have to do this in the embryonic cells. Fine. After gene mapping, you have to take those cells. It's going to be highly effective. If you can only do it in embryonic stage, not in adult stage. It's a statement given in your NCERT. It's a statement given in your NCERT. It's a statement given in your NCERT. Clear? Just check it out. Just check it out. It's a statement given in your NCERT.
Sir,
Yes.
কোনটা বুঝতে পারবো না স্যার।
কোনটা?
বোন ম্যারো।
আদা
বোন ম্যারো বুঝতে পারছি। এক সেকেন্ড। বোন ম্যারোটা বুঝতে পারছি।
আদা বুঝতে পারছি.
স্যার লিম্ফোসাইটে চলে তারপরে
আদা
Okay, bone marrow.
আচ্ছা মেয়েটার নাম মেয়েটা কালো অ্যাকচুয়ালি সেটা হচ্ছে কি
কালো জিরে.
অন্য ম্যানি কোন নাম আদা কোনো মিশিয়ে মানে আরে আদা হলো তো রসুন.
Okay, bone marrow.
কালো জিরে. আদা কালো জিরে.
কালো জিরে. এটা সাজেস্ট করতে হবে এটা আমি
Okay. Fine. Where is the problem?
স্যার
Yeah, Khanda is the problem. Okay. Tell me. Fine.
স্যার CD এটা লিম্ফোসাইটের জেনারেশন মানে
Introduce Colli by the help of retrovirus. Now lymphocyte is loaded. Initially it was not having ADA. Now it is having ADA. Happy or unhappy?
প্রবলেম স্যার আছে এসি যা
Happy? You have grown in culture medium, huge numbers. Then put it inside. Introduce these cells inside bone marrow. Inside bone marrow. Bone marrow is the original copy. Remember the Chatu speech in 3 Idiots. Virus. The Chatu.
সিনেমার কথা বলো সবাই.
Yes. No. Wait. I need to tell you something. The Chatur क्या किया था उसने? चतुराई से चतुर में रैंचो
চেঞ্জ করেছিল ওর
चेंज किया था ना कुछ. He changed some of the words, some of the letters. He changed some of the letters in the original copy. Original copy में ना. और वो उसका वो क्या लिखा था? Copy लिखा था ना. Original is what? Bone marrow cells are the original cells. दे आर द स्टेम सेल्स, ओरिजिनल सेल्स. उसके अंदर आपने क्या इंट्रोड्यूस कर दिया? नहीं, lymphocytes. हाँ, what kind of lymphocytes? Having ADA or no ADA? Having ADA. So ADA दिखाते दिखाते वो काम किया ना, नहीं किया? क्या काम किया? Because you have introduced it inside the bone marrow cells. It is going to produce now. It's going to produce what kind of cells? The cells having loaded with ADA gene. It's going to be effective now. But ये किस में किया आपने? एडल्ट में किया या एम्ब्रायनिक स्टेज में किया? ये लिखा हुआ है आपके बुक में, निकालो. लास्ट लाइन ऑफ दिस जीन थेरेपी. लास्ट लाइन ऑफ दिस जीन थेरेपी. लास्ट लाइन ऑफ दिस जीन थेरेपी. क्लियर? लास्ट लाइन ऑफ दिस जीन थेरेपी. देखो यहाँ पे. See? Okay, let me tell you one thing. I missed one point. All of you listen to me very, very carefully. Fine. Just wait. If this was found in a 4-year-old child, ADA, the 4-year-old child is not having the defense system, so he was kept under a bubble, a coating, where, because in this air you have got virus, bacteria, every other pathogen. So if you are a 4-year-old kid without ADA, so you are going to die, because all this bacteria is going to get inside your body. So उस बेबी के चारों तरफ there was a bubble formed, and that's why this baby was called as, this particular disease is also called as bubble baby disease. Bubble baby disease. Bubble, it's a, no, there is a covering artificially made, just like space suit. A covering made so that the virus or bacteria cannot invade the body. Think about it. Otherwise, you are not going to be helped. You will not. বেঁচে ছিল? See, till the time his body is going to get this ADA. So think about it. This is what. Okay, if a person is born, hey, all of you, listen to me. देखो जल्दी से. Now to crack your exam, these are the informations which are needed. If a person is born with a hereditary disease, gene therapy is an attempt taken for correction of such disease. Gene therapy is a collection of methods that allows correction of gene defect that has been diagnosed in a child or embryo. Okay? Here genes are inserted into a person's cells and tissues to treat a disease. Correction of a genetic defect involves delivery of a normal gene into the individual or embryo to take over the function of and compels to form a non-functional gene. Okay. And our first clinical gene therapy was given in 1990 to a 4-year-old girl with ADA, adenosine deficiency. Need to run 16, 20, and 23. The next one, 26. ADA deficiency causes SCID, severe combined immunodeficiency. In SCID, B lymphocytes and T cells are not formed. Think about not formed. मतलब suddenly India में सब soldiers they are become very much motivated by one of the person. I'm not talking about the person. You know that chal chena chal bena. So they stopped working. They stopped working. Think about what is going to happen to your nation. Think about what is going to happen to your nation. Bolo samajh mein aaya ki nahi? So you're going to get vulnerable. Now ADA is crucial for the immune system to function. ADA is crucial for the immune system to function. The site of production of ADA in the body is
lymphocytes.
Which cells forms ADA normally? Fine. In your body, what is the cell which forms ADA and shows ADA? Lymphocyte cells. Got the point? Okay. In some children, RDS can be cured by bone marrow transplantation. बोलो. Bone marrow transplantation. ज़्यादा कॉस्टली नहीं है. Google. Fine.
17 crores.
Okay. 17 crore. As a first step towards gene therapy, lymphocytes from the blood of patients are grown in a. As a first step, very, very important. As a first step towards gene therapy, lymphocytes from the blood of the patient are grown in a culture outside the body. Culture outside the body. কাকে grow করা হয়েছে? The lymphocytes are being grown outside the body. In vitro or in vivo? In vitro. Okay. A functional ADA, cDNA, using retroviral vector. What is cDNA? If you introduce this gene of the ADA inside retrovirus, it's going to change it into DNA. That is called cDNA. That is called cDNA. Is then introduced into these lymphocytes, which are subsequently returned to the patient. देखो, उसके बाद patient के अंदर दे दिया आपने. And you know, very very important. सिर्फ retrovirus ही काम नहीं करता है. Also, adenovirus is also one which can perform this one. Okay. Just wait. I missed one particular point. Remember, retrovirus having that particular gene of ADA, when infects a lymphocyte cells, it is done by an enzyme called integrase enzyme. Write it down somewhere. Integrase enzyme is there with retrovirus. What is the virus? Integrase enzyme is the name of the virus.
Sir, within retrovirus.
Within the retrovirus. Is there with the retrovirus. What is the name of the enzyme? Integrase is the name of the enzyme. Integrase is the name of the enzyme.
The use of this.
How?
Use of this enzyme.
It integrates the foreign DNA with their DNA, the ADA DNA with the lymphocyte cells.
Or DNA ligase.
DNA ligase. Ligase is always there inside the cell. But this integrase is a special one which is there inside retrovirus. Adenovirus vectors is used to replace the defective gene in gene therapy. This question came in 13. Very, very important. So what is the other virus? Apart from retrovirus, there is adenovirus is used to replace the defective gene in gene therapy. However, as these cells are not immortal, the patients requires periodic infusion of such genetically engineered lymphocytes. बोलो समझ में आया क्या? As these cells are not immortal because lymphocyte cells का लाइफ स्पैन क्या वो immortal है या उसका लाइफ स्पैन है कुछ? है? सोल्जर्स का लाइफ टाइम होता है एक बार घुस गया इंडियन आर्मी में बस 200 साल से काम करेंगे हम नहीं रिटायरमेंट होगा नहीं होगा सोल्जर रिटायरमेंट होगा तो उसके साथ क्या चला गया आड़ा जीन भी चला गया lymphocyte cells मर गया आफ्टर इट्स लाइफ स्पैन इज ओवर सो lymphocyte cells লাইফ স্প্যান lymphocyte cells লাইফ স্প্যান হচ্ছে খুব বেশি হলে হয়তো দেখিস ক্লোজ টু 10 to 15 days 10 to 15 days yeah it's good because that is the time span for it to work efficiently समझ आया क्या अब बूढ़े सोल्जर को लोगे अब सोचो with the adipose tissue like me so enzyme replacement therapy in this method functional ADA is given to the patient by injection कोई लाभ होगा enzyme replacement therapy में कोई काम होगा functional ADA is given directly to the patient by injection but the problem with both of these approaches is that they are not completely curative temporary effect है इसका however if the gene isolate from the marrow cells producing ADA is introduced into the cells at early embryonic stages, it could be a permanent solution. So what is not being given over here is about germline and somatic cells. कहाँ पे ये होगा sure shot? Germ cells में देने से, not in the somatic cells, not in the somatic cells. Clear, Babu? Any problem? That's all about gene therapy. Let us complete the chapter today. Fine. Molecular diagnosis. In this topic called molecular diagnosis, for effective treatment of the disease, early diagnosis of understanding pathophysiology is very important. The study of altered physiology of the host is called pathophysiology. Using conventional methods of diagnosis, serum and urine analysis, and early detection is not possible. The technique that serves the purpose of early diagnosis is PCR. We have done it. RNA technology. We know that. ELISA, we know that. In ELISA, what happens? Antigen-antibody reactions. See in the year 2021-23. Presence of a pathogen, bacteria, virus is normally suspected only when the pathogen has produced a disease symptom. By this time, the concentration of pathogen is already very high in the body. have a very low concentration of bacteria or virus at a time when the symptoms of the disease are not yet visible can be detected by amplification of the nucleic acid by PCR. I think you know what is amplification. That what is done in COVID. COVID ki kora hoyechilo. What happened in COVID? RT-PCR. How you have detected that the small genome. You have got 6.6 into the 9 base pair, and the COVID virus was having roughly approximately 5,000 base pairs. So, at the lomba DNA মধ্যে এইটুকু জায়গা ছিল virus. So how to detect that particular portion? By amplifying it by the process of PCR, and that's why RT-PCR. Why RT being done? What kind of virus was it? Was COVID virus? It was RNA virus. So you have to do reverse transcription. PCR is now routinely used to detect HIV in a suspected AIDS patient. PCR is also used to detect mutations in genes in suspected cancer patients too. PCR is a powerful technique to identify many other genetic disorder. In cancer, what is going to happen? Some of the genetic base pairs are going to change. They don't have PT dimer. So how to detect huge amount of PT dimer? By amplifying it. PCR করে কি করবে? এটাকে amplify করবে, and you want to have huge amount of DNA. A single stranded DNA or RNA tagged with a radioactive molecule is called probe. We already have done radioactive probe. Come on. In DNA fingerprinting, we have done probe. Yes. Probe is allowed to hybridize to its complementary DNA if in a clone of cells found by detection using autoradiography. Baba, we have done this in DNA fingerprinting. If you have not watched the live video of DNA fingerprinting, you are going to get all the information. The clone having the mutated gene will hence not appear on the photographic film because the probe will not have two times. Okay, two times. Complementarity with the mutated gene. Okay. Genes of interest can be selected from a genomic library by using DNA probes. ELISA is based on the principles of antigen-antibody reactions. Very, very important. NEET 2020, 23 and 26. Okay. Principles of antigen-antibody interaction. Infection by pathogen can be detected by the presence of antigens. CCC. What happens in ELISA? Infection by pathogen. Baba, foreign body क्या है आपके लिए? What is foreign body for you? Adito alone at home and some foreign body.
Foreign body.
Foreign body. So Adito is having two options. One, by opening the door, is going to say Baba boleche, Baba nahi. Yes. So that person asked for his father. As the open korlo, Baba boleche, Baba nahi. So what he has shown? Antigenicity. I think we have talked about this. He has shown antigenicity. What is the meaning of antigenicity? Who is the foreign body antigen? और उसने क्या किया? आने दिया बॉडी में? नहीं. क्या शो किया? Antibody. Stop the person. Antigenicity. What is the next option? It's the second option. The moment he opened the door, found that person. नहीं. एक स्टिक लिया और दौड़ना स्टार्ट किया मारने के लिए उसको. मार ही देगा उसको. This is called immunogenicity. This is called immunogenicity. So all immunogens are antigens, but all antigens are not immunogens. Most of them. Okay. So infection by pathogen can be detected by the presence of antigens, proteins or glycoproteins, or by detecting the antibodies synthesized against the pathogen. अगर body में उसका antibody produce हो गया, that means you are having what? If your body is having the antibody of AIDS virus, that means you are already having what? आपके body में क्या घुस गया? Antigen घुस गया, and that is called ELISA. Okay. Very, very important topic: transgenic animals and plants. Transgenic animals and plants. You know what is called transgenic animals? We are not talking about that. Yes. What we are going to talk about? We are going to talk about, transgenic models exist for many human diseases such as cancer, cystic fibrosis, rheumatoid arthritis. It's an example of autoimmune disease. Alzheimer's. Tell me.
The frontal lobe.
Alzheimer's produced due to deficiency of dopamine or acetylcholine. Alzheimer's, dopa. Acetyl. Check it out. Alzheimer's. Biological product. Easy action. Okay. Very important. Is the biological product.
হ্যাঁ তো ভুলে যায় যে ডোপামিন
ডোপামিন
তো হ্যাপি
Tell me.
হ্যাঁ দেখেই বুঝে গেল
Serotonin.
ডোপামিন তো হ্যাপি হরমোন চলে
Good,
যাচ্ছে
good,
বলতে
good,
গেলে।
good. There is some neurotransmitter. Dopamine is also a neurotransmitter. Acetylcholine is also a neurotransmitter. So what is common? These two are common. Neurotransmitters are dopamine, adrenaline, and acetylcholine. No? Richa na?
Which question Alzheimer's which hormone লিখে দেন
Charging.
Okay. Baba, come over here. Biological product. Biological products. Let us talk about a particular cow.
লেট
Particular.
এলিচ কি
No. Alzheimer's नहीं भूलिए.
No single hormone
No,
directly
no.
causes
Alzheimer's.
Alzheimer's.
Alzheimer's में सिनेमा देखते थे
Alzheimer's.
দাদু আছে
Find out. Find out.
কেরলা সিনেমাটা আরে
Schizophrenia.
একটা নতুন
Schizophrenia.
Alzheimer's মানে
আহান পান্ডে
Sir.
হ্যাঁ না
Sir.
Alzheimer's. Nerve disorders. Nerve disorders. Rosie the cow. See, who is the cow? Rosie. Baba. Rosie. In 1997, the first transgenic cow named Rosie produced human protein. What is the name of the human protein? Alpha lactalbumin. So the gene.
no single hormone is responsible for schizophrenia.
No, it's not hormone. It's not hormone. Neurotransmitter.
And dopamine.
Dopamine. Schizophrenia.
Serotonin.
Dopamine.
GABA.
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 Yes, they are neurotransmitters.
Glutamate.
Schizophrenia.
Sir, acetylcholine.
For what?
Alzheimer.
Alzheimer's, acetylcholine. How to remember? A for Alzheimer's, A for acetylcholine.
ইজিটা হবে না
And schizophrenia, dopamine. Okay, Baba, let's come back. Hey, Baba. Now, Rosie. देखो रोजी एक गाय है जिसके रोजी एक गाय है ट्रांसजेनिक काउ. What is being introduced?
Rose.
No. What is being introduced? The gene which can make human alpha lactalbumin. So बोलो हर एक प्रेजेंट डे मदर जिनका मिल्क प्रोडक्शन नहीं होता है बिकॉज टू द लाइफस्टाइल चेंजेस. इतना अच्छा लाइफस्टाइल है.
Seriously?
So they are going to make Q. लाइन बनाते हैं रोजी. Why? Because that Rosie is having the gene of what? Human. So ये रोजी क्या नॉर्मल काउ है या ट्रांसजेनिक काउ है? So yes, got the point? Okay. Now the milk contains the human alpha lactalbumin and was nutritionally more balanced product for human babies than natural cow milk because cow milk is for calf. It's not for human babies. Come on. Okay, now, what is this? There's another one which is called alpha-1 antitrypsin. This is a particular protein to treat emphysema of lungs. And what is emphysema of lungs? I think you know that. Chedi Singh, very good, Chedi Singh. Remember that? The alveoli, they get linked together and the person who smokes that nicotine activates that enzyme and you know what happens with the two alveoli? There is a gap which occurs between two alveoli and there is a pore which forms inside the lungs which is called emphysema of lungs. Emphysema of lungs can be treated by introduction of portion of a DNA or genes which codes for a particular product such as human protein. What is the name of the protein? Alpha antitrypsin. Alpha-1 antitrypsin. Important. Okay. Vaccine safety. We are not going to go line by line. You just study that. Chemical safety. Yes, chemical safety. Remember some of the animals who are very much sensitive to some of the chemicals. Some of the deodorants. Foreign company when they try to introduce deodorants or a perfume, they first to check what kind of chemical is present. They put on the eyes of this guinea pig having transgenic one. Fine. If they lost their vision, this particular this particular chemical is not being allowed to get marketed in India. So that is chemical safety testing. Chemical safety testing. Ethical issues, very, very important. Ethical issues. There is a committee called GEAC. Think about Aditya is having all the restriction enzymes and all the genetic tools in his home or in his research lab. Do you think Aditya can do whatever he likes? He can make whatever test he would want to make? No. He has to take the approval of GEAC. Fine. Say for example, Aditya has made his. His means snake with human head.
What?
A snake's body with human head. इच्छाधारी नागिन में ये नहीं था. It was in a particular movie called His. Now, think about it. Okay, Baba. Now, so can he make this? He's having all the tools.
Yes, he can.
Yes, sir.
বানিয়ে ছেড়ে দিলে তারপর কে কি বলবে?
No. He never knows that his is going to first hiss on him only.
স্যার ও তো মানুষের মাথা তো কি করে হিস করবে?
আচ্ছা তো উল্টো করে দে.
জিমটাই
Change it.
সাপের মতো বাবা
কিরকম লাগে?
না. Got the point? সাপের মাথা, human body.
কি বাবা
Now think about it, Baba. So the point is, he has to take the approval from GEAC. Clear? Which makes decisions regarding the validity of GM research. Okay. What is biopiracy? What is biopiracy? Very, very important. See, the use of bioresources by multinational companies and other organizations with proper authorization from the countries and people concerned without compensatory payment. See, compensatory payment. So you can take, say Aditya has made some of the formula. So I am from American company, I've taken the particular formula from him saying, आपको पैसा मिलेगा. और उसके बाद मैं कभी लौटा नहीं इंडिया. This is what? Biopiracy. I've taken the formula from him. Never gave him any compensation for that, any payment for that. He was having this patent. अब ये patent का पैसा मैंने नहीं दिया उसको. बोलो समझ में आया कि नहीं आया? हाँ? Very good. So NEET 2018-2021. Rice is good. Rice is an important food grain, the business of which goes back thousands of years in Asia's agricultural history. There are, Bishina, 2 lakh varieties of rice in India. Alone. The diversity of rice in India is one of the richest in the world. Okay. Basmati rice is distinct for its unique aroma and flavor. 27 documented varieties of basmati are grown in India. Terms are important. 27 documented. Fine. There is a reference to basmati in ancient texts, folklore and poetry as it has been grown for centuries. Mama, I think this is yes, basmati. Think about it. So in 1997, so very good thing by America already because they create all this and they break all this. Fine. In 1997, an American company got patent rights on basmati rice through the US Patent and Trademark Office. This allowed the company to sell a new variety of basmati rice in the US and abroad. This new variety, got the point? घर में बोलिन है ना? मम्मी को पूछो, ये बोलिन का स्मेल functions में कोई चेंज हुआ for the last 40 years, 50 years? Patented formula. This Bengal chemical product is a patented product. इसका कोई आप there was a product named Boro Calendula. Marketing करने के लिए कोशिश किया था. Wanted to fight with Bolin and wanted to take the market share of Bolin. Bolin अभी भी आपके next generation उसके बाद next generation चलेगा Bolin. Fine. अभी आ रहा है ना दिन लगाने के लिए. Vaseline में कोई Vaseline is not going to repair it. वो सिर्फ ऊपर कोटिंग देता है. बोरो प्लस क्यों?
লিপ থেরাপিটা ভালো
লিপ থেরাপিটা ভালো আর ভ্যাসলিন নতুন এসেছে আমি এনেছি
Okay, Bolin is a patent product. Okay. Basmati had actually been derived from Indian farmer varieties. India basmati was crossed with, hey, listen to me. India basmati was called the semi-dwarf varieties, right? Semi-dwarf varieties and claimed as an invention or a novelty. Acha, tell me, semi-dwarf variety, neither too tall, neither too dwarf. Semi-dwarf variety of this paddy field is good or bad? If it is going to be long enough, tall enough, কি হবে? What is going to happen? They will droop down. Fine. So inka lateral semi-dwarf is going to have lateral branches, more of lateral branches, going to hold more of rice, more of paddy. The patent extends to the functional equivalence implying that other people selling basmati rice could be restricted by the patent. आप कैसे sell कर रहे हो ये? So restriction हो जाएगा. Several attempts have also been made to patent uses, products and processes based on Indian traditional herbal medicines. Example, turmeric, neem. The Indian Parliament has recently cleared the second amendment of the Indian Patents Bill that takes such issues into consideration, including patent terms, emergency provisions, and research and development initiatives. Got the idea what is called patent? So इसको बायोपायरेसी रोकने के लिए आपने पेटेंट बिल किया. Otherwise पेटेंट मैंने ले लिया था आदित्य से. Who is earning in crores? Me. और what about आदित्य? 2 पैसा. एक रोल दे दिया उसको. अच्छा बोलो. Any issues with this?
Yes.
The transfer of genetic material from one bacterium to another through the medications or vector like virus is termed as transduction. We know it. I will tell me transduction के लिए name the 2 scientists who made that
Zinder
experiment.
and Hindenburg.
Zinder and Hindenburg.
Ginger. Ginger.
I'm not talking about that. Name the 2 scientists in your class 12. There is a test performed by 2 scientists named Hersey and Chase. Hersey and for transaction. Think about it. And that is Zinder and Hindenburg. Availability of thermostable DNA polymerase has popularized the PCR. Extension of primer and end of the template's DNA is catalyzed by a tag polymerase in PCR. Calimulus, important. Calimulus was awarded the Nobel Prize for the development of PCR technique. I have talked about this in PCR. Calimulus, I have talked about him. Baba, that's all about. I think that's all about the gene therapy. Just let's start with one of the questions, 1 or 2 questions maximum. We are going to do it. हो गया? Audio. Just one question.
Question नहीं खाने.
We have done it already last class.
नहीं खाने.
Silence in our mind. We have done it. Okay, that's all about this chapter. Biology is over. Biology and its applications is over.
Thank you, sir.
Okay, thank you. Thank you. So next day we are going to start with a new topic. Okay? So that's it. Sure. हाँ कौन सा?
मूवीटा
कौन मूवी है ना?
आप सामने